Hi everyone, so my name is Noor. I'm one of the 3 PALTiers for Dr. Harrington's section. And this week what we're going to be talking about is PCR and Sanger sequencing. And so I will do a little bit of a background for both of these concepts just because you actually really need that background knowledge in order to answer these questions. And so to make sure we're on the same page, I will do that review. However, if you already understand and know the concepts of PCR and Sanger sequencing, you can just skip right to the part where I start talking about the questions. Please keep in mind that this is my first time filming a video like this for a worksheet, so it's not the best setup, but this is all I had to work with. And also, just last thing is to please try this worksheet before you watch this video so that you're able to figure out the questions on your own. And then if you have problems understanding something, then come watch this video. So to start off, I'm going to do a review on PCR. So what is PCR? So PCR is the process in which we are going to be amplifying or making millions of copies of a certain DNA fragment. So if this was your entire chromosome, the thing is when you're doing PCR, you're not actually amplifying your entire chromosome. You're only amplifying a certain piece within your chromosome. So usually what you would want to be amplifying is your gene of interest. Okay? And so what PCR actually does to actually make a lot of these copies of DNA is to actually just go through DNA replication multiple times. So in order to do that, we need 3 things in our reaction tube. We're going to need our DNA, of course, our template DNA. But we're also going to need primers, right? Because DNA polymerase needs a free 3' end to actually extend from. So we definitely need those primers. We're also going to need a DNA polymerase enzyme because that's the enzyme that's actually going to put the complementary nucleotides onto the new strand. And finally, we're going to need nucleotides, right? DNTPs, which are just deoxynucleotides. So, in order for us to actually do multiple rounds of replication, the first thing we're going to want to do is denature our DNA. And so, what denaturing the DNA is going to do is it's going to give you access to the DNA strands, right? So just like this. The second step, so this is step 1. So you're gonna probably do this at about 95 degrees so that the DNA strands are going to separate. The second thing that's going to happen is a step called annealing. And so what's happening in the annealing stage, is that your primers are now going to actually anneal to your DNA strand. So if this is your denatured DNA as before, this stage what's going to happen is your primers are going to bind to wherever you made it complementary to, right? So if we go back here, if this is our gene of interest, we're gonna want to have our primers pretty close outside, but outside of our gene, right? So that we can include the entire gene that we want. So in this case, here would be our gene, and so you're gonna design your primers to be right next to your gene of interest. In addition, just to make sure we're very clear on this, so if you guys remember from the beginning of the course, we reviewed DNA replication, and maybe you remember, but we talked about that DNA is made 5' to 3', right? Which just means that you start with a 5' end with your primer, and then DNA polymerase is going to be adding on to this 3' end, right? So if this is your 5' end, because imagine here, this would be your left primer, so it's going this way. So if DNA is being made this way, this would be your 5' end, and that would be your 3' end of your primer, right? So if this primer starts with the 5' end here, because DNA is always made anti-parallel, that would mean that this strand, this side of the strand, is the 3' end. While this over here, towards this way, is going to be the 5' end. Similarly, if this is your right primer, it's going to be going that way, right? And so if DNA replication is happening in this direction, then what's going to happen is this would be your 5' end, because again, DNA is always made 5' to 3'. And so if that is your 5' end of your primer, like this would have to be the 3' end of your DNA, so that it's anti-parallel. Okay? So the process of annealing, which again, just to reiterate, is just the process of which the primers are going to anneal onto your DNA strand. This is going to happen at a slightly lower temperature of, 55 degrees, not 72. And then after your primers are going to anneal, what's going to happen is you're going to want to actually add these complementary strands, right? So you can actually have new molecules of DNA. So this step is going to be called extension. So all that extension is it's the process in which your DNA polymerase is now going to be adding your complementary nucleotides to your strand to make brand new DNA molecules. So if this was your DNA as before, and here's your primers. DNA polymerase is now going to be going in that direction because we established before that this was our 5' and this is the growing 3' end. And then same thing here, because this was your 5' end of your primer, your DNA polymerase is going to be extending onto your extending your 3' end. And so the process of extension is going to happen at a slightly higher temperature of 72 degrees, and that is because your the DNA polymerase that we used originally is actually known as Taq DNA polymerase, and Taq DNA polymerase functions optimally at 72 degrees. So the reason that we actually have to do these 3 different temperatures is first because denaturing DNA has to be at a really high temperature. Primers usually only, they anneal at certain temperatures depending on how you design them, but when you optimally design them, they're usually around 55 to 60 degrees, and then you have to raise the temperature again up to 72 degrees because that's where DNA polymerase functions best. Okay? So that was the process of PCR, and so as you can see, once the DNA polymerase is actually done replicating these 2 strands, what's going to happen is you're going to repeat this whole process over and over again, and that way, because you're going to be taking these new DNA molecules and replicating them, and you're going to do that about 30 to 40 times, you're going to end up with a lot of DNA, which is what you wanted. So hopefully the process of PCR makes sense to you all now. And now I'm going to spend a little bit of time reviewing Sanger sequencing as well. So this is automated Sanger sequencing that we'll be discussing today. And basically when you're sequencing DNA, as you can tell from the name, what you're trying to do is you're trying to determine the sequence of a certain DNA molecule. So if this was your DNA, and you want to actually sequence this molecule. You can either sequence a certain gene or the entire genome. It's usually up to you how you design your primers. But anyways, what you want to actually start by doing when you're doing Sanger sequencing is you're going to want to actually amplify your DNA. So you're actually going to do the process of PCR first to actually generate, I'm just like making them smaller, but imagine those are the exact same DNA molecules, because you want to generate a lot of your DNA. And this will become clear as to why a little bit later on when we actually get through the process of sequencing, yeah. So once you've actually amplified your DNA, what you're going to do is you're going to denature it so that you can have access to it, the DNA molecule. And so what you do when you're sequencing DNA is that you're actually going to also design primers to anneal to a certain part like that you want to actually sequence. So for an example, if you were looking to sequence this part here, then what you're going to do is you're going to design a primer that is going to be right next to that sequence, right? And so the thing though is that unlike PCR where you use left and right primers, in this case you're going to be only using a left or a right primer, not both. Again, the reason will be clear in a little bit, but you only use either a left or a right primer, not both. PCR uses both primers to actually amplify the DNA. Okay, so you're going to need primers. And so your primer is going to come in and anneal to your denatured DNA. And then you're going to have DNA polymerase come in, and it's going to add DNTPs, which are just nucleotides, right? So in this reaction mixture so far, what we have is DNTPs, we have primers, and we've also put a DNA polymerase, right? So DNA polymerase is going to start adding the complementary bases to this DNA strand, right? Except we don't only add regular nucleotides to this reaction mixture. We also add these nucleotides known as ddNTPs. And what these ddNTPs do is they're going to stop DNA synthesis. So wherever a ddNTP is placed, DNA synthesis from that point on no longer occurs. So if the DNA polymerase is randomly, this is going to all happen randomly, by the way, where like your ddNTP is going to be placed anywhere depending on whatever the DNA polymerase picks up and puts, right? So if this is your ddNTP, DNA polymerase can no longer add any more nucleotides to this molecule. So it just stops here, okay? And the thing is that these ddNTPs are also tagged, or they have a fluorescent dye attached to them. So for an example, the adenosine nucleotide may have a green fluorescent dye attached to it. Similarly, thymine might have a blue, for an example. G may have a, this is not the primer, these are nucleotides. Sorry, I should have probably done it in another color, but G may be purple, and C may be pink for these ddNTPs, okay? So let's just say that this, on the original strand, we had a C, then the ddNTP that's going to be complementary to that is a G, and this G is going to have a purple fluorescent dye attached to this ddNTP. Okay, hopefully that makes sense. So again, what's going to happen is because you have so much of this amplified DNA, the DNA polymerase is going to do this in multiple times. So it's going to put ddNTPs randomly throughout the sequence. So what could happen is one of your strands could be 2 nucleotides long, depending on where the ddNTP was placed. If it was on the second nucleotide, then it's going to be 2 nucleotides long. But it could also put it 10 nucleotides from this primer. And so you're going to have a 10 nucleotide long fragment of like newly synthesized DNA, right? And so that's the reason why we had to actually amplify all this DNA, because what we want to do is we want a ddNTP to be placed on every single one of these complementary strands for our complementary bases for each one of these strands. So hopefully that kind of makes sense. So what you end up having then from this process is DNA of different lengths. Some will be much longer than others, again, just depending on where the ddNTP was placed. So you could have 2, then you have 4, and then you have however many. So you're going to have one of these for each of your bases. And that's what you're going to use to actually figure out the sequence. So what ends up happening after this is you have this machine that you place all of your mixture into, and you put it into this negative electrode. And the thing is because DNA is negative as well, it's going to be repelled by this, and it's going to be attracted to this positive electrode. So DNA is going to flow in this direction. The thing though is that within this machine, what it ends up doing is it's going to actually let the smaller DNA fragments, so for an example, if a DNA fragment was 4 nucleotides long, it's going to reach this end first. And what we have here at this positive electrode is a fluorescent dye detector. So you're going to have 4 nucleotide long DNA, then right next to it you're going to have 5 nucleotide long DNA, and then 6, and so on and so forth until you reach, the end of your sequence. And so then what's happening is when this fluorescent dye detector, when this DNA molecule comes in, is it's going to detect whatever fluorescent dye was attached through that ddNTP. So for an example, if this is pink, we said before pink is C, then they know, okay, if this ddNTP, sorry, this is a little bit, but maybe I wouldn't use this example, sorry. But essentially, if it knows that a C was placed as the final ddNTP, because that's the one with the fluorescent dye, then what we know is if a C was placed here, so I'm going to actually erase, if a C was placed here, then they know that the original strand had a G, right? Because if it detects a C, which has the fluorescent dye attached to it, then they know that this would be G, okay? And so what's going to happen is all of your DNA, from smallest to biggest, is going to come in, and it's going to run through this machine, and you are going to be able to determine the sequence of DNA based off of that. So hopefully that made sense. I know that it's a little bit confusing, but if you're still confused, you can watch probably some videos online on YouTube or something. And just one more thing now that I think about it, so that you guys understand that as well, is that, again, so DNA is made 5', so if this is your primer, it's going to be made 5' to 3', right? So whatever is coming through this machine, the first couple of like DNA molecules are going to be closer to the 5' end of your new DNA sequence, and then your new strand is what I mean. And then towards the end is those that were going this way, right? So near the 3' end of your new strand. So hopefully that makes sense. I'm sorry if I was very confusing. What we're going to do now, though, is we're going to move on to the worksheet for this week. And hopefully this will make, when we do an example of it, it will make more sense. So this question tells us that to recall the start codon is AUG, and you are amplifying a wild type gene using a left and right primer. And then you're going to be sequencing your PCR products that you got through amplification, and you're either using the left or right primer. Okay? Again, because sequencing either uses a left or a right, not both. So, part A is asking us to, maybe I'll, oh, you guys are zoomed in. Okay, it asks us which of the above shows what the start codon would look like if you sequence with a left primer. Okay, so to actually start off this question, it's super important to remember the start codon, right? So they told us the start codon was AUG. So AUG, right? This is 5' to 3' because mRNA is made 5' to 3'. However, what's really important to remember is that when you're sequencing DNA, you're sequencing DNA, not sequencing mRNA. Okay? So actually, before you even start this question, you know that you can eliminate 1, 2, 3, and 4. And the reason for that is because, again, we're not going to be sequencing this mRNA. We're sequencing the DNA that encoded for this mRNA. Okay? So you can automatically get rid of those 4 options to begin with. The next thing that you want to do is, because, as I mentioned, we're sequencing the DNA, we're going to want to know what was the template which made this mRNA. What was the sequence of this template strand? So, the template strand that made this mRNA is going to be anti-parallel, right? And it's going to be complementary to this strand, to this mRNA. So if there's an A here, that would mean we have a T in our DNA, template DNA. If this is a U, that would be an A. And if this is a G, this would be a C. Okay? So that is what's important to recognize. And then what you can also do is, now that you have the template strand, you can use the non-template strand, or this complementary strand of DNA, and just find out what your DNA sequence is for the double strand, as you can see here, and then that's your mRNA, right? So you're just finding what this strand now, the non-template strand sequence was, and it's just going to be complementary to this. So it's going to be anti-parallel, and it's going to be ATG, right? To be complementary. Okay, so that's the first thing that you want to establish when you're doing this type of question. If they give you an mRNA and you need to figure out the DNA, you want to do this. The next thing now that you need to consider is what direction of the primer are you using. So in this case, it's the left. And let's look up here for the left primer. What's happening? So the left primer is going in this direction, this way, right? And so if DNA then is going to be built in this direction, that means we started with the 5' end, and we're building on this 3' end. So that's our newly synthesized DNA that the sequencing machine is doing, right? That you're doing for sequencing. Now, in order to, I'm just gonna get that. So this is our new strand. So then our original DNA, our template strand, is going to be anti-parallel to this, right? It's going to be 3' to 5', right? To be complementary to that. So that's our newly made strand that's going to be used for sequencing, and it's going in this direction. And so if you actually look then, what we want for our template strand is on the left side to be the 3' end and the right side to be the 5' end. And if you look right over here, that would be this strand here. We're using this as your template strand when you're using the left primer. So in that case, if your template strand is TAC, then the machine, as it's going 5' to 3', your machine, as it's making the new 5' to 3' strand, is going to be adding whatever's complementary to this. So it's going to be A, then it's going to add the complementary nucleotide to the A, which is T, and then G. So that is what's going through your machine, right? Because if you remember what I was saying earlier, was that you are detecting the strand that you are making, not your original template, because your new strand that you're making is where the DNTPs are going to be added. So that is why for this case, you would have number 5 as your correct answer, because it's going to be adding the complementary to this template. So hopefully that made sense. Now looking at what would the start codon look like if you used your right primer, well, your right primer is going in this direction, right? So if your right primer is going in that direction, then that would mean this would be the 5' end, and this would be the 3' end of your newly synthesized strand. So if that's where your primer is annealing, it's going this way. So if it's going this way and is 5' to 3', to make it anti-parallel, it has to be made anti-parallel. This original DNA strand would have to be 3' to 5', right? 3' on the right side and the 5' on the left. So if we go back now to our sequence that we would be using, now the template strand, when we're using a right primer, would be this one over here, right? So here it would be G, T, A, right? That was what you would see on your new DNA. That was what you would see on your template strand. And so what's going to be now added complementary to this, your newly synthesized DNA, which is the one that's going to have the fluorescent dyes, is going to be complementary to this original strand, which will be CAT, right? And so then in this case, the C is going to be detected first because that's going to be the smallest strand made, then the A, then the T. So in this case, number 8 would be the answer for that. So hopefully that made sense. It's definitely a really tricky question. Not tricky, but I guess challenging is the way that you would word it. But hopefully now that you've had practice with this question, if you see this question in the future on your final or something, you'll be able to actually understand how to do it. So now we're going to move on to question number 2. Question number 2 tells us that Huntington's disease is an autosomal dominant, and it's due to mutations in the HTT gene. And there is a CAG repeat in the middle of the protein coding region. And so if you have 35 or fewer of these repeats, so if you have 35 or, yeah, like this, yep. If you have, I'm not even, I forgot my math. But if you have 35 or fewer repeats, then your protein is functional. While if you have 40 or more, then your DNA is your protein will not be functional because there's too many glutamines. And so what we want to do in this question, it says, is to describe how you determine a patient's genotype using a PCR-based procedure. Okay, so let's think about why we would be using PCR in this case. So if you think about PCR, and what we're doing is we're amplifying our DNA. And so to actually look at a patient's genotype and to discern whether they have a mutant or a wild type allele is to look at how many repeats they have, right? So if they have if they have 40 or more, then they would be mutant, but if they have 35 or less, they would be wild type, right? And so what we can do is we can actually take our DNA and we can run it on a gel, if you guys remember that concept. And what the agarose gel does is it's going to separate the strands based off of its size, right? And so the smaller ones will be towards the bottom, while the bigger ones will be towards the top. And so basically, the reason why we're using PCR is because we want to amplify our DNA and have enough DNA so that we can put it in our agarose gel, and also because we specifically want to look at the HTT gene, we want to specifically amplify that gene. Because if we were to take the entire chromosome 4 and run it on a gel, there may not be enough differences between these two, like when you have such a large chromosome, differences of 5 or 7 codons is not going to really show up on your gel. But if you amplify specifically this HTT gene, then you're going to actually be able to see those observable differences. So that is the reason why we're using PCR. Now, to actually list our steps of PCR, the first thing you're going to want to do is you're going to want to obtain this person's, your patient's DNA, right? So you're going to obtain blood, and you're going to want to obtain blood from patient. But they also tell us that we need a control, and this is super important. Why? Because we need to compare somebody with a wild type, which is 35 or fewer repeats, to someone, we need to use that to compare, right? Because how would we know the size of a patient with a mutant allele if we didn't know what the size of band for those that have a wild type phenotype? You need just like someone to compare it to, right? So you're going to want to obtain blood from patient and someone with wild type alleles, right? Okay. The second thing is once you've obtained your blood, this blood has a bunch of random things like cells in there. So what we want to do is we want to actually isolate our DNA from those people, right? Isolate the DNA from the blood. Step number 3 would be that we would want to have primers so that we can specifically amplify the HTT gene. So we're going to design and order primers. Step number 4 is once we have our primers, we have our DNA, we're going to want to actually do the process of PCR. So we want to set up our reaction mixture, right? And in our reaction mixture, we're going to have our template DNA. We're going to need our primers, right? Left and right. We're going to need our DNA polymerase to actually go in and add those nucleotides. And then finally, we're going to want to have nucleotides to actually be adding for the DNA polymerase to add. Step number 5 would be now, once you've had this reaction mixture, is to actually run on PCR. You run your PCR and a thermocycler, because if you guys remember, I mentioned before how PCR happens in 3 steps that occur at 3 different temperatures. Finally, the last thing that you want to do is you're going to want to take these PCR products, thermocycler machine, and you're going to want to take those PCR products and run them on a gel to look at the differences. So run PCR products. On a agarose gel from the 2 reactions, the person that was wild type and from your patient. Okay, now in part B, they tell us to draw the results if the patient does or does not have the mutant allele. So, this would be, for an example, this would be our agarose gel, and we're going to need 2 lanes, right? One for our patient and one for our wild type person to compare to. So, let's say wild type and patient. And let's just say in this case, the person does not or does have the mutant allele. Let's see what we would expect. So if the person was wild type, what that means is they have 35 or fewer repeats in the protein, right? And so on a gel, when we run this, the larger fragments are always towards the top, while the smaller fragments are always towards the bottom. So in this case, somebody that has the 35 or fewer might be somewhere around here, right? And because this person is wild type, or like let's just say that they were, in this case, homozygous dominant plus, or homozygous recessive, because this is an autosomal dominant disease. So this person would be autosomal recessive with 2 wild type alleles, right? Now in this case, if the patient has a mutant allele, that would mean that they would definitely have a dysfunctional copy of the HTT gene, but they could also either be homozygous dominant because it's an autosomal dominant disease, or they could be heterozygous. So let's just draw the scenario where they're heterozygous. So if this person is heterozygous, what we'd expect is, because they have 1 copy of the functional allele, they would have a band at the exact same place as person number 1, right? Because they have 1 functional allele that has 35 or fewer repeats. But because they also have this dysfunctional allele, they would have 40 or more CAG repeats. So because our agarose gel goes from smallest to largest in terms of size, we would expect a band to be higher, right? Because they are going to have they're going to have more repeats, and therefore their DNA sequence is going to be longer than the person that was wild type or recessive for the HTT mutation. Okay. Let's draw if the patient does have, or doesn't, sorry, we just did doesn't have mutant allele. So let's say this is our agarose gel, 1 and 2. 1 is still the person that's wild type and so has 2 functional alleles. But now patient doesn't have our mutant allele, so they're actually going to have the exact same genotype as the wild type. And so then what you can imagine on our gel then, what it will look like is that the wild type again will have 1 band, and then our patient, because they have the exact same genotype, is going to have the same band in the pretty much the exact same place, just depending on how many repeats. But yeah, they would have a band in the same region because again they don't have that mutant allele. So that's how we could use PCR and agarose gels to actually determine the genotype of a patient. So, moving on to question number 3 now. Question number 3 tells us that the A gene is 5 kilobase pairs long, and that the A+ allele has a restriction enzyme cut site 2 kilobase pairs downstream of the promoter, while the mutation A- changes this sequence to this, right? So that was our original cut site sequence, and now this is our mutated one. And so the thing is in this, when the person has a mutant allele, what ends up happening is this restriction enzyme can no longer recognize this sequence, and so then what's going to happen is the DNA fragment won't be cut. So that's giving a little bit of a hint for like the rest of the question. But to start off with part A, it tells us what kind of mutation is. So let's actually compare these two sequences for our DNA. So here we have a G and a G, that's the same. But here, our second nucleotide is an A in the wild type case, but in the mutant case, this person has a T mutation. And so we know that it's a base pair substitution, right? Because the A is changing to a T. So it couldn't be like a nonsense or missense, I don't know about missense, sorry, or nonsense, never mind. Ignore what I just said. But we know it's a base pair, so it's not an insertion or deletion. That's what I meant to say, sorry. So we know that it's a base pair substitution because the A is changing to a T. So it's an A to T. And now, usually though, when we're writing mutations, what we want to do is that we want to not just include A to T, we usually include the complementary strand. So if this was our sequence, GAA, TTC, and this is our wild type, the complementary to this would be CTT, AAG, right? And then, but if they have the A- mutation, then instead what we get is GTA, TTC. And so what's complementary to that would be CATAAG, okay? And so if you look, what we're actually really having is an AT, is our original AT, and it's changing into TA. And so that's just usually the way that we write our mutations when it's a base pair substitution. Okay? Part B tells us that after that, what you're going to do is you're going to isolate your DNA, set up a PCR reaction, digest the PCR products with the restriction enzyme, and then run on an agarose gel. So it tells us to make a diagram of this experiment by drawing the mutation primers, PCR products, and restriction fragments. Okay, so I guess we can start off by looking at the wild type allele. So the wild type allele is not going to have a mutation, so we don't need to draw that. Let's go in and draw the primers. So the primers for a, sorry, just 1 second. I just want to make sure I'm drawing it the right place. So primers are usually going to be drawn just right outside your gene of interest so that you can include your gene of interest. So it would be somewhere right around there. Hopefully you guys can see that. Maybe I'll zoom in. Okay. In addition to that, it tells us to draw our PCR products. So our PCR product is going to be this gene amplified, and a little bit of where the primer and yield, but that's not important. So that's going to be our PCR product, and our PCR products are DNA. And then it tells us to draw a restriction fragment. So in this case, because we have the functional allele, we know that our DNA is going to be cut, right? And in the beginning of the question, they tell us that the EcoR1 cut site is 2 kilobase pairs downstream of the promoter. So what we'd expect is a DNA fragment that is 2 kilobase pairs long, and if the A gene was originally 5, then 5 minus 2 is 3.